Good morning. I think we should get started. So it's my pleasure to introduce Dr. Jonathan JIrish, who is an assistant professor at Vanderbilt University in the Department of Cancer Biology. And I'm very happy Dr. JIrish agreed to come to talk to us. I'm a big fan of his body of work in the area of what I would consider translational pathology, and what I would like translational pathology to look like. Essentially, it comes down to high-dimensional analysis of cells, both in solution and in tissues, to get as much information as it's possible out of every cell. And what happens then? You end up with way more data than you can swallow. So Jonathan was also instrumental and is the founder of an online analysis platform for analysis of high-dimensional data called Cytobank, which is used by thousands of researchers worldwide. And his interest is in cancer biology and applying those methods to improve outcomes in cancer. So, without further ado, I would like to ask Jonathan to give his presentation. Thank you.
Thank you very much for the opportunity to talk here. So that's a great introduction. I'm thrilled that you think of us as translational pathology because that's what we're aspiring to. I'd really like to help pathologists someday. So all of the work in my lab starts with human tissue. And with live cells from tissue biopsies. And I trained in immunology, but now I'm moving in my lab at Vanderbilt into solid tumors and solid tissues. And so I'll talk about that here, the challenge of doing single cell work with solid tumors. But the idea is that we want to get all of the cells, make as many measurements as possible, come up with not only what the cells are expressed, but how they react to things, do functional assays, see what they can secrete, measure cell signaling. And by monitoring and dissecting how the cells behave during treatment in people, we can make the treatments better. And so the overview for the talk today is I'm going to introduce the idea of cytomics, which I'm starting to think of as the omics of cell identity. It means cell omics, cytomics, but I think it's the figuring out all of the cells that are in a tissue and characterizing them. And so it's one of the few omics that has many different platforms that can do it. You can measure both DNA, RNA, protein, metabolites, functional responses to characterize a cell. And so I think it's kind of special that way. I'll go into a case study of systems immune monitoring. I'll talk about how we're adapting techniques for solid tumors and tissues. And then I'll spend a large amount of time on a new computational technique we created called marker enrichment modeling. And the idea here is that we want to make it as easy as possible for a computer to identify cells. And so I introduced this idea of cytomics, which is going to be characterizing all the cells that are in a primary tissue. And I think that this field really got a boost with the advent of mass cytometry, which is a high-dimensional version of flow cytometry that I'll talk about a lot more. And so in 2011, Sean Bendel, Aaron Simons, and others from the Nolan Lab published this science paper where they took healthy human bone marrow and they created a flow panel that was about 35 markers, and it measured features of all the cells from the hematopoietic stem cell down through the differentiation tree to capture the myeloid, lymphoid lineage, and everything in between. And so with one flow panel, they could recapture computationally all the cells in hematopoiesis. And so this paper has had a huge impact on the field and on me. In 2014, Evan Newell's group did a mouse version of the same idea where they made a mass cytometry panel to capture myeloid and innate lymphoid cells and took 8 mouse tissues, brain, kidney, spleen, bone marrow, and so on, some of them solid tissues, and characterized the entire myeloid immune system in the mouse. And so this is what we mean by cytomics, characterizing all the cells in a tissue or all the cells in a system. And I made this kind of analogy in this Nature Immunology News and Views with Evan Newell's paper, where we're going from this old map where there are regions that we might think of as cyto-incognito, the unexplored territory, where there are cells we never even knew existed that fell between our gates or had really unusual phenotypes, or cells that we were reluctant or afraid to explore, like microglia in the brain. And so I think that with mass cytometry and high-dimensional flow, we're revealing all of these. It's like we're sending up satellites to map the Earth, and we have this really comprehensive system, but we're also getting a little bit of distance. And so we need to design our assays well to ask questions about the cells. Because with 35 markers, it's still not that many. You still need to focus your question and ask a choice. And so this is from a review article that we put together, characterizing some of the different types of mass cytometry panels that we build of about 35 markers. And so you could make a panel that focuses on all of the cells in a tumor microenvironment, but that limits your ability to go after intracellular readouts. And so on the left would be trying to track every type of cell and get deep into subsets. And on the right would be sort of the opposite extreme of measuring phosphoproteins or cleaved caspase 3 or something inside the cell to characterize the signaling network of the cell. And in that case, you're going to have a limited number of markers for the major cell types. And then in between, we might try and capture cell-cell interactions for something like a checkpoint inhibitor therapy that's designed to block those, or pick one cell type, Tregs or something, and do a really deep immunophenotype into them. And the goal of this work is to maximize the knowledge gained from each patient biopsy, just like was said in the introduction, and ideally to learn from the in vivo, because if we sample blood or even tumor from patients who are being treated, we can learn a lot about how the treatment works, maybe during treatment as well as after treatment. And this is really being powered by technology advances. So it's kind of a joke. So we're going all the way from the 1600s with Hooke coining the term cell by looking at the structures that looked like jail cells. And so this is kind of a low-dimensional approach where you grind your own lenses and build your own microscope and then characterize the tissue. But so flow cytometry has really been powering this. And I think it's kind of a maxim in the field that every dimension we add multiplies exponentially the number of cell types or cell subsets present. And so in the mononuclear phagocyte system, CD14, which is part of toll-like receptor 4, was really definitional in identifying classical macrophages. But as soon as they added CD16 and FC receptor, you can see it fractionates the cells. So we have CD14 positive, CD16 negative classical monocytes, and then we have non-classical, and then we have intermediate that express both. And so every marker that we add starts war among immunologists arguing about which cells are the real cells and whether they're part of it, and trying to define which cells do a particular function or are important for a clinical response. And so with mass cytometry, when we went up to 32 dimensions and could do 500 cells per second, this inherently revealed new types of cells that people hadn't seen before. And this is a common part of the field. And it generates this flood of data, as was said. And so we need computational tools that can cope with all that data and ideally identify or at least give us a hint about the identity of the cells automatically as a first pass part of the analysis. And so most of the work that I'll show today is mass cytometry data. We still do a lot of fluorescence flow cytometry, and we go back and forth between them. So fluorescence flow is really high throughput for cell number. You can run thousands of cells per second, whereas mass cytometry is more like 500 cells per second. And so you have to sit there for almost an hour in order to get 1 million cells. But on those 1 million cells, you can measure much more than is traditionally capable with fluorescence flow, and there's very little spillover between the channels. And so if you don't like compensation, mass cytometry is for you. And this is because, so on the left is fluorescence flow, and on the right is mass cytometry, and the fluorophores that are used as probes in fluorescence flow have a lot of spillover into each other's channels. And so you detect them, and there are a lot of clever ways of dealing with this, but in mass cytometry, the peaks are very sharp. And we have a mass window of about 100 to 200 theoretical isotopically pure heavy metals that we can detect as our probes in mass cytometry. And so instead of a fluorophore conjugated antibody, we have a polymer on an antibody, and the polymer chelates an isotopically pure heavy metal like gadolinium-156, becomes our reporter. And so this is something that's not naturally seen in a cell, and the cell picks up a bunch of the antibody and picks up the gadolinium as a result, and then we stream it into the argon plasma. There's no cell left at that point. It gets atomized and ionized. There's not even molecules left. And then we filter the low mass ions, run the heavy metals along this track, quantify them, and that's how we can measure the abundance of the targets. And it has a fantastic dynamic range. It's not quite as good as PE, but it's better than FITSI, for those of you who do fluorescence flow. And so I've called this cytomics, and it's not just me. Other people use this. So this is a Nature Immunology review, and I was really pleased to see that the cytome made it into the list of omics and ohms on the thing, so it's part of all of these. But I think what's kind of maybe special about it is that for genomics or epigenomics, there's usually one type of technology or platform, but for cytomics, you can blend lots of different types of information. You can even do imaging, single-cell imaging, and use positional information as one of the ways of characterizing the identity of the cell. And the Bowden-Miller lab has really taken the lead on doing some of that. And I've used this term cell identity, and I'm trying to stay away from terms like lineage, because I've stopped thinking of cells as having very stable, irreversible lineages and started thinking of everything as polarization and gradients of identity and how firmly held or weakly held is a cell's identity. And if you want to read more about this, there's a nice review by DuPage and Bluestone where they go into this idea for T cells, and they say maybe we should think about them as polarized along different spectra and acquiring different functional capabilities, but moving between those polarization states. But then there are other identities like B cell and T cell that don't appear to interconvert. And so we're obsessed with this idea of cell identity, and we want to do it for human cells. And so what I want to be able to do is take any tube of human cells that you hand me, run a mass cytometry panel or set of panels, and be able to cope with any cells that are present in there. And immunology can do this today, but as we've started to move into other types of tissues, so we wanted to do glioblastoma, and so we're trying to do neural cells and melanoma as well as neural crest origin, we realized that other fields don't have this single-cell protein-based identification system without morphology and positional information. And so I think there's a real need in these other fields to come up with something like that, to look at the cells from the immunologist's point of view out of context and try and identify every single cell. And then there are some types of cells like fibroblasts that are sort of defined as leftover after everything else. I mean, they have strong morphological features, but if you put them into single-cell suspension, it's really hard to identify fibroblasts, or there are many different types of fibroblasts, and they're sort of tissue-specific. And so I think that there's a lot of work still to be done to characterize what are the singular features or defining features of different cell identities, and how do cells move through those identities, which transitions are allowed and which aren't. And so these are, I'm highlighting Vanderbilt papers here. This is very self-centered, and so these are, this is our work and our collaborators out of Vanderbilt. There's lots of other great labs doing this type of work and doing fluorescence flow and other things, but this is a mass cytometry list. And if you're really interested in this stuff, we'll have AI and AACR, if you're at those meetings, we have some posters on lung cancer and melanoma and glioblastoma that show some of our very latest work that I won't have time to get into here, but you might want to go grill the people at their posters about some of these. And so I think of cancer and the immune system as competing cellular systems that are changing over time, and so this ability to track individual cells is really powerful because you can see how the system of cells is changing, how their identities and functional properties are changing. And so we come up with kind of a general hypothesis that if we model all the cells before and after treatment, we might reveal biomarkers, and ideally these biomarkers might be mechanisms of therapy response or therapy resistance. But as I sort of said before or hinted at, chance favors the prepared assay. What you choose to measure is really important here, and this is what we spend a lot of time losing sleep over. Are we measuring the right things? What is going to reveal the key cell subset or the key property of those cells? So some of the markers that you'll see throughout my talk, I'm going to focus largely on surface markers. In this talk, it's not as much of a cell signaling focused talk, although that's a real interest area of mine. And so throughout the talk, you'll see markers of differentiated cell types. And so there's a quiz later. I know this is for continuing medical education. And so this is great. There's an actual quiz built into the talk that you'll all take at the end. And so you can think of the hematopoietic stem cell as CD34 positive and CD38 low or negative. This compartment is enriched for stem cells, and these cells lack CD45. And then as the cells differentiate, they gain CD45. They specify into these different cell identities, and T cells, for example, have CD3 and might have CD4 or CD8, and K cells have CD16, and you'll see these markers throughout the talk. You'll see HLA-DR or MHC class II in humans, CD14 and 16 on macrophages, and then B cells are well known for CD19 and CD20. So either you know that or you've all just memorized that, so good job, and it's about 20 slides later, so hang on to that. So this is the case study. So this is a mass cytometry data, and I've plotted it so that it looks like traditional flow cytometry data. So we can see dots, the y-axis, dots for cells. The y-axis is CD45, the x is CD33, HLA-DR, and CD38, which are three things that can be expressed on myeloid lineage cells. And so this was a melanoma patient who had previously, decades earlier, had breast cancer and been treated with radiation, was back for melanoma, and was being treated with anti-PD-1 checkpoint inhibitor to try and activate the immune system against their melanoma. And we were sampling the blood to try and do a systems immunology study to look for biomarkers of response to anti-PD-1. And so this is a story about finding something unexpected. And so we were definitely not expecting to see myelodysplastic syndrome, MDS blasts, in the blood of this patient, but that's what we saw. And so on the left, you can see a healthy donor, and in this sort of region, there are not a lot of cells that are low for 45, so immature cells expressing CD33. And then you can see this small population of blasts that's expanding over time. And we don't think this has anything to do with the anti-PD-1. We think that this is just something that was going on in the blood of that patient, but it's something that we could then track using mass cytometry and look at all of the other markers that were present in our panel. But what was striking to us was that the standard CBC actually missed this at the pretreatment. And I don't know if this is normal, and this is probably totally unfair, but the CBC is really bouncing around a lot here. And this purple line is our mass cytometry quantification, and this is, if you have a graduate student pore over the data for hours, and it's part of their, PhD thesis. So it's pretty unfair to compare a standard of care CBC to a graduate student poring over the mass cytometry data, looking through every marker for something. But it was striking that we were able to track it, and that a rare population like this isn't missed in this type of panel. So the other thing about these mass cytometry panels is that we have all the markers in one tube, so we can break down all of the different cell types, track them, and then look at expression of different markers on them. So we can pull out, for example, the MDS blasts here and track their abundance, and then for the other cell subsets, we can now track and see that they were declining. And surprisingly, the non-MDS myeloid cells, or the cells that didn't look like the blasts, monocytes, for example, were also sort of taking off. And so as the blasts were taking off, we saw diminishment of the other immune compartments, but the myeloid compartment really expanded. And we can now go back and ask, was PD-1, for example, a target on the blasts? Did we uncheck the blasts and let them expand or something like that? And we don't think that that happened. But if we had done this with fluorescence flow, we probably would have had PD-1 in our T cell panel and not in our myeloid cell panel. We would have had to sort of sacrifice. Whereas we had everything here, so we could go back and look at PD-1 expression on all the different major cell subsets, and we could dig down into different finicky cell subsets. But there's not really a story here. There was a little bit of PD-1 expression on some of the myeloid cells, but they and the blasts were not very strongly positive for PD-1. But the main point here is that we couldn't have done this with our 10D staining panel design. So as we started to do more of these longitudinal systems immune monitoring studies where we have a hypothesis, but we also want to be open to seeing all different types of cells, we've tried to come up with a new analysis workflow that won't miss anything. And so my postdoc mentor used to say I have FMS, fear of missing something, and he's absolutely right. And so we've tried to think about what are the major things that you would want to get in an automated computational analysis. And so abundance or change over time in the frequency of cells is a key one. So we just saw that. The plasticity or stability of the system is one that our group and others have been using something called Earth Movers Distance to characterize how much the overall system is changing or staying the same. I'm not going to talk about that at all today, but Petter Broden's lab has some really cool talks about that I think you can find online. I will talk a lot about this tool, MEM, or marker enrichment modeling, that we've developed to try and do the last two parts of this, which are those cells new, and what are their signature features? What makes that cell subset special that I found? And we realized that this was really the hard part. This was where you had to train a graduate student and have them spend several years of their life learning the literature and then poring over the data to figure out what was new and special about the cells. It was much harder to get a computer to do it automatically. So before I dive too much into it, I'll also say that we've spent a lot of time on techniques, and we're now moving these into a core-like setting as a service, and the idea is to really ramp up all of the mass cytometry, especially the immune part of it, which is really robust and standardized, into a fee-for-service environment. So we just started this, and I think that this is a really helpful way to imagine mass cytometry going, that there will be two parts of the community. One group that's the expert users who want to access the machine, are designing their own panels, creating new panels, or pushing it into new technology, and then other groups who want to do it in maybe a multi-center immune monitoring setting and really want things to be standardized, robust, not have to worry about batch effects or things like that. And so I think if you're putting it into a core setting, you might want to think about how are you going to support those two very different types of users. And so we've set this up as a total fee-for-service type of thing where people who are doing this can't even touch the machine. So they just hand a tube of cells to somebody, that person has the antibodies, stains it, collects the data, and so on. And there are things like data analysis and training and experiment design that are part of this. And so this is our protocol for solid tumor and solid tissue disaggregation. And this is published now in a Cytometry B article last year, and we're working on a current protocols article that'll come out really soon. But if you've done a FICOL prep of blood or bone marrow, it's very similar to that idea. So the key thing is you just need to digest away the extracellular matrix. And so I'm trying to convince more people to store viable single-cell suspensions from more tissues. That's what we really need. The dead cells don't signal. They're not as interesting. You can't make cell lines out of them, although there's a lot of things that you can do with fixed tissue and imaging. But I think the cell signaling and functional responses are relatively understudied. And so we take a surgically resected patient sample. Usually it's a person goes to the operating room or somebody in the OR, drops it into PBS, keeping it at room temperature, covers the sample, and then they bring it to the lab. We sort of chop it up in a macro way with a scalpel, mincing the tissue, and then we spin it down, discarding the supernatant, incubating it at this step for about an hour with collagenases. Then we filter away the large particles, lyse out the red cells and platelets, which is really important to do well, and then we can viably cryopreserve the cells. And we developed our cryopreservation protocol both for viable cell yield, but also for cell diversity. We wanted to make sure not to lose different types of cells that are present in the tissue at the beginning. And so we sort of went back to the drawing board and evaluated all different conditions that I'll show here. And we used tonsil as a control because you can mechanically disaggregate tonsil really easily. And we thought, oh, of course, we won't be able to improve our tonsil prep protocol, but we did. And then we compared glioma, melanoma, and we have some small cell lung cancer xenografts in the sample. And so one of the things we do is we always take a picture of the cells on a hemocytometer so that we can see what they look like and count the viable cells early on. And so our viable cell counts is really old school. It's just looking at a hemocytometer and asking how many viable cells per gram of tissue did we get. And we found for glioblastoma that the collagenases that we tried, there was no one collagenase that was the killer collagenase that gives you really good yields from all different tumors. Some tumors gave better yields across collagenases. Some tumors gave lower yields, but they were all sort of within the same ballpark. It was really important to have DNase present during the single cell release, and ideally you kind of want it throughout the assay. So dying cells that release DNA, it sort of acidifies and kills other cells in the tube, and if you're thawing cells that have a lot of platelets or that are dying, you might want DNase in when you're thawing them as well. And so you can see no DNase, and with DNase we got many, many millions more cells released. So that's glioma. Melanoma was a similar type of story where it was really valuable to both have collagenase to digest away the extracellular matrix and DNase. And I didn't say trypsin is bad. So it's bad in a couple ways. So it's going to cleave extracellular antigens that you want to use to detect the cells, but it's also, the cells need those things to stay alive, and so it seems to be killing the cells. And so we haven't had great luck with trypsin or other things like that. And so this is tonsil. We were a little surprised to see, so no enzyme is what we've been doing for a long time. When we added collagenases in some samples, we released hundreds of millions more cells than we were getting without collagenase there. And as I've gone back and thought about it, these might be other types of cells, fibroblasts, epithelial cells, that we seem to be missing normally in our tonsil preps. And so we haven't even had time to go back and characterize what are these extra cells that we're releasing. Are they just more B cells and T cells, which is normally what you get with this protocol? But I'm optimistic that they might be some more interesting cells. So this is some of the timing optimization that we did with different collagenases. And the graduate student was previously doing 3 hours, and because there was this thought that longer is better. We're going to release more cells the longer we go. And that was definitely not the case. So shorter time points were better, and we think about 45 minutes to an hour is about the ideal time point. But the key thing wasn't, we just didn't want to maximize for viability. We wanted to maximize for getting all the different types of cells that are present in the tissue and not losing anything. And so this is an example with a glioma tumor where all of the dashed lines are terminal gates that you're going to see on the next slide, where we're going to quantify those cell subsets over time. And so the first population is cleave caspase 3 positive cells, so apoptotic cells. And then we take everything that's negative for that, and that's positive for histone H3, which is going to be a nucleated cell marker here. And then we call the CD31 positive cells endothelial in this case. And this is the old school biaxial gating way. We don't really do much of this in my lab anymore, and I'm going to show you Visny and other computational tools. But I kind of wanted to walk through how we could do it and then track them on the next slide to orient you if you're coming from traditional flow cytometry gating. And then the CD45 positive cells are the immune cells, and then everything else will be on the bottom row. So within the immune cell gate, we have MHC class 2, HLA-DR, for antigen-presenting cells that were all CD64 positive. And so in brain, these are, we think, essentially microglia. And then within the CD3 compartment, this is a part of the T cell receptor, and so those are T cells, and we can see, we can call them memory or not based on ROL. That's a little broad. And then within the non-immune, non-endothelial compartment, we have alpha SMA, and we called that parasites, CD24, and we called those ependymal cells. And then within this negative gate, we can do SOX2. And so SOX2 is, it's one of the Yamanaka factors, can induce pluripotency, helps keep cells stem-like. And we think that these might be really interesting cells within these glioblastoma tumors. They might be either neural stem cells or a stem cell subset of the tumor. And then there's this glial fibrillary acidic protein, GFAP, an intermediate filament, which also can mark stem cells in the brain and is positive in the glioblastoma tumors. And then some other markers that, if you're into this field, are part of it. So S100 beta, which is also in melanoma, and the PDGF receptor. So we subsetted all of these subsets of cells within the tumor, and then we tracked, do we lose them over time during our prep protocol? And so you can see the cleave caspase 3 up on the left here. We're getting more of these, and we don't want to get more of those. These are the dying cells. And so some of the striking things to me are we're losing microglia, and we worked really hard to make sure they're not adhering to plastic or being lost because of that. Similarly, memory T cells, so this is the 45 RO positive, but not the RO negative cells, were lost. I don't know why that is different. I thought that was kind of interesting, because they're both T cells and both fairly robust. And then you can see some other, our favorite population, like the SOX2 positive cells is sort of plummeting. Out here, there's some SOX2s over, sorry, these are the negatives, these are the positives. They're dropping off really early. This GFAP positive population lasted overnight and then was lost. And then at the end, this is kind of the gate of everything that's left over, and these are the ones that are making it until the end. So I'm going to skip my animations there. And so, but you can see that traditional hand gating approach is completely impractical and sort of biased to what we already know. And there's always this question of where do you draw the line for the gates, and there are some cells that fall between the gates. And so we need computational tools that can look at the data and not lose track of any of the cells. And two of my favorite tools are Visney and Spade. So Visney was published by Donna Pear's group in this Nature Biotech article. Spade came from Peng Chu in another Nature Biotech article. And so Visney is a tool that takes your cells and spreads them out in 2 dimensions according to their the protein markers you choose, according to their phenotype for those proteins or other features. So similar cells will be placed in a similar part of the map. Spade is a clustering tool that creates clusters of cells based on phenotype. And so this is some melanoma data, and if you're, I'm not going to talk at all about our melanoma story, but I'm happy to talk more afterward. Dion will be showing some very updated melanoma data at AACR if you're interested. I did want to show just a little bit of glioblastoma data to kind of orient you to Visney maps before I dive into them and explain them. And so this is a Visney view of glioblastoma where we've drawn this map. So each dot is a cell. We've drawn the map using protein features of cell identity. So we have CD45 for immune cells, HLA-DR for antigen-presenting cells, and then TUJ1, a neural marker, Nestin, SOX2, or other glioblastoma cell identity markers. And so, and this is not, it kind of looks like a tissue microarray, and some of these end up being sort of round when they don't have a lot of structure, but it's not. So this is not the original architecture of the tumor. The cells have been placed in this map so that phenotypically similar cells are close to each other and form these sort of islands of cell density. And so down here, you can see these are the antigen-presenting cells, the microglia that were present in that particular brain tumor. And you can see sort of islands of differential expression of Nestin or EGFR within the tumor compartment. And one of the best things about Visney maps is that every cell that you put in gets a spot on the map, even if you don't know what it is. And so it gets related to other cells by its closeness or proximity, maybe touching some of those other islands. You can get some hints about what it is. But we also, so Visney maps are great, but we have to do this thing, what we've all been doing, called stare and compare, where we look at them and we kind of look which marker is up, which marker is down, and we've wanted to move away from stare and compare and get automated tools that might sort of pop up a label and say antigen-presenting cells over here. And so that's the goal of MEM, and that's where we're trying to get things to. Okay, so how does Visney work? So this is how Visney works at a high level, and then I'll show a little bit of an example. So this is, we have to cite our sources. This is the Cloud Cuckoo Palace instructions from my son's Lego set. And so we were really excited when we did mass cytometry, but we got overwhelmed. And so in this analogy, the Legos are cells, and their color and number of pegs is sort of the cell phenotype. And what Visney does is it essentially just moves the cells around in 2D to create little piles out of them that are the different phenotypic islands of cell identity. And then we can go in and try and figure out what those islands are. And so what it actually does is it looks at the cells in the original high-dimensional space, and it calculates a similarity score for how likely a given cell is to pick another cell as its neighbor. How similar is it to another cell? And it does this for every cell versus every other cell. And then it scatters the cells into 2 dimensions and progressively moves them around and asks, is their 2-dimensional position close similarity score close to their high dimensional one. So this is the start of the animation, and you can sort of imagine we're zooming out, and every frame of the animation, it's moving the cells and asking, is there similarity score? Are the cells in this region, were they originally similar to each other in high dimensional, 35-dimensional space? And so what we find at the end of these maps is that if we backgate from two-dimensional views, that this is the monocyte island, this is the CD4 T cell island, CD8, natural killer cells, and B cells. And they get put here based on the markers that we measure, like CD19 and CD16 and so on. And right now you have to take my word for it, that these are the monocytes and these are the CD4 T cells. You can kind of guess based on the abundance, and this was healthy human blood, but MEM is designed to put a label on this that tells us empirically what was differentially enriched in this blob of cells versus the other ones. Because I got sick of looking at other people's VISME maps and SPADE maps and having to take their word for it that this little thing was hematopoietic stem cells when it looked like some junk off to the left there or something. And another thing that happens with VISME a lot is that if you backgate, so this is from Donna Pear's original paper, and this is Evan Newell's Nature Immunology article, and they backgate expert knowledge where an expert tries to find all the cells that they are comfortable with. There are always these gray islands that peek through. And these gray islands can take up about half of the sample in some cases. So they say here 54%. And there were 3 very clearly structured islands of cell identity within there that the expert had overlooked. And so they basically got a Nature Immunology article figuring out what these 3 populations are and showing that they change when they knock out the GM-CSF receptor. But really, they got it for comprehensively describing this myeloid immune system and revealing that there are cells that we had no idea about. And there are these cells, like I said, that kind of go between our gates or that are up in the corner, and we thought it was junk, but really they have a defined phenotype. So in 2015, we published this bioinformatics workflow that we called a modular workflow, and we sort of went through how our lab does the data analysis. So we start, and this is now a course with a step-by-step workflow. You can download the data. We're teaching courses on it all around the world. If you're interested, we're very happy to share data sets and step-by-step protocols to start with the exact files we do here. So we usually do a major cell gate. We run VISME to spread the cells out in 2 dimensions. We do SPADE, and we've started to cluster or do SPADE or other tools on the T-SNE axes. And this is fraught with peril. The computational biologists have explained this to me many times, but it also works really well. And we can talk more about why it might be a good idea or a bad idea, but we're exploring now whether we should be clustering on these 2-dimensional reduced views or in the original space, and there's some value to using both pieces of information as well. But what you choose to cluster on is really important, but you need to put the cells into populations or groups at some point, and then you need to characterize those groups. And in this article, we were characterizing them with heat maps and looking at clinical features, but now we're using them. And we made the point in this article that so humans must still stare and compare and use expert knowledge to identify the populations at the end of it. And that populations, we kind of give them these metaphors. We might say, oh, central memory T cells. But are my central memory T cells the same as somebody else's central memory T cells? Or if I just use PD1 to mark CD8 T cells, are there a million other things in my cytoph panel that are different about those versus somebody else's PD1 positive CD8 T cells? And so we've gotten really interested in this idea of an unbiased way to label and identify cells and share knowledge. And so as flow cytometry people, we're used to staring at lots of cell distributions, and we realized that we were largely comparing median as a way of capturing a degree of expression. Is something highly expressed or low expressed? But when a human looked at it, we were also taking into account how tight the peak was or how broad it was. And we would say, even with the same median expression, that if the interquartile range, the IQR, is lower, that is enriched. It's more enriched, or maybe even more important to that cell's identity than when it's spread out like this. And so the underlying idea of them, the sort of assumption of it, is that population distributions that have smaller interquartile range are more enriched for that feature than other populations. And so we created a really simple equation where we take the log difference in the median and add to it the ratio of the interquartile ranges of a reference to a test population. So a test population might be CD4 T cells, and the reference population might be all the other cells, or it might be hematopoietic stem cells or something that we want to compare it to. And we ask for each marker, like CD4, is the interquartile range smaller in the test than it is in the reference? And if it is, then this number will blow up and get bigger. And if they're the same, it'll be 1, and so we subtract off 1. And so the mem label will be 0 if the population has the same expression and variation in that marker as a reference population. And so, and then we transform these to a -10 to +10 scale. I'm kind of a Dungeons and Dragons nerd, and so that's part of where that comes from. We just think of it as a 20-point system and how many points you get for a given marker. Negative numbers mean that the marker is specifically missing from the population. 0 means there's no difference, and positive numbers means that it's specifically enriched. And so it creates this label where it organizes that information from the most enriched to the least enriched in the positive direction or the negative direction, so that you can actually kind of look at this label and read it. And if you know that CD20 and CD19 are key identifiers of B cells, you might be able to just look at that label and say, that is a B cell population. And so now it's quiz time. So we have 8 labels here. And can you tell me, so the percentage of this cell in the tissue is listed in the corner, and the label is there. The easy ones are on the right if you want to start there. Can you tell me what any of these populations are and what this tissue is? And I really hope that you can. And don't worry, they're really easy populations. Does anybody want to take a guess?
Top left corner is myeloid blast. Let's see. Next one down is...
This is healthy, by the way. So, yeah. Top right looks like NK cells, definitely.
Next one down after top right is cytotoxic T cell.
Yep.
And then next underneath that is helper T.
Yep.
And then under that is T cells.
T cells.
And then over to the left, bottom left is those who are myelomonocytic.
Yep.
And then up above that. And
so I'm going to pop up the labels. So these are the labels. Good job. So these are the labels. This came from Sean Bendel's paper. Yeah. That's great. So yeah, they called this population hematopoietic stem cells. And this came from their spade tree. So I'm going to click to the next slide. These are the same labels.
I would have called them myeloid stem cells because they express CD3.
Yeah. So now we can kind of get into arguing about that, because they just took this orange circle and said that was HSCs.
Whoa.
Yeah.
Who are they?
And so we can ask, but I thought this label was pretty good. I mean, it had 34 and it didn't have CD45, and it wasn't a B cell, you know.
It's purposefully vague, which is what pathologists are supposed to learn.
And we've also learned, some artificial things about our cytop panels. So if there's the equivalent of a compensation problem, if there's a metal from the wrong channel spilling into a population, this kind of picks it up and reveals it to us. And so we don't mean to say that the mem label is truth. It's just an empirical observation about what was enriched in that population. It's like a median or a fold change or something. And so we can overlay these on the tree. The reference really matters, and so we explicitly take into account a comparison point. And so for this one, each of these major sort of bubbles was compared to all of the other cells in the tissue. So that's one way that we typically do MEM. Another one is we pick a common reference point. So here, this is the same data set represented a different way, where we used that HSC node and compared all the other nodes all the other populations they identified to it. And we get these enrichment. This is the heat map of the MEM scores, so the -10 to +10 score for how enriched something is in a population. And you can see, for example, CD45 is coming on in the differentiated cells. And so the context really matters. And so if we take the exact same CD4 T cell population and compare it to all the other cells in the tissue, and this is one that we got out of tonsil, or if we compare it to a stem cell reference that we took by combining multiple people's mass cytometry data on a gate they called hematopoietic stem cells, we get two different labels. So compared to other tissue cells in tonsil, CD4 T cells are lacking in CD19 because most of the other cells are B cells, but compared to an HSC, they're very highly expressing CD4 and CD3. And so I think immunologists sort of naturally think of an HSC or a cell that's sort of negative for everything as their comparison point, and we find that these sort of versus the gold standard labels are the more readable, but these are really useful for characterizing signature features. Like if you wanted to design a gating scheme to find the cell, you might want to use the versus other cells in the tissue. So I'm going to go a little more quickly to get into some of the other examples. So this is an example from blood where we did, so this is the T-SNE map. We identified these major populations just as a proof of concept. And you can see like CD45 for median expression is very hot, but it's not different at all among these blood cell populations. And so if we look at the variance across them, the variance is incredibly low, but it's high for CD3 and CD4 within healthy blood. And so the MEM label for CD45 is 0. It's not different for any of these populations, but it's going to be very low or very high for the things that are the most variable within that tissue. And so you can see some of the other MEM labels for healthy blood cells on the left there. So do these features matter for clustering, and do they distinguish real cell types? And so do they matter for clustering? This is the y-axis here is F measure. Sorry, it's not labeled, but it's a 100% down to 0 in the F measure. And what we did here is we took the markers that MEM found as highly enriched in cells, and we took them away as we clustered using VISNI. And we asked, do we get the right cells in the right clusters of populations? And so if MEM is identifying good markers, we'll break it really quickly as we take those markers away. And so the F measure will decrease really quickly. If we remove markers randomly, that's this top curve here. And so you can see right away MEM separates out. And by about 7 markers, we've gotten down to 70% is this first line. And so then if we remove them based on median expression, the highest expressed markers aren't the most useful for defining population identity because some of them, like CD45, don't vary at all. And then we also used like Kolmogorov-Smirnov or Z-score as a way of identifying differentially expressed features, and this still wasn't quite as good as MEM. And so a graphical representation of that on the left, so this is a VISNI map, these are the same. If we exclude markers by high expression or by enrichment, you'll see the map changing. So this is the good map, and we're now backgating NK cells and CD8 T cells. And so you can see as we take markers away from MEM, we sort of destroy the map more quickly than when we take them away by median. So we've taken away the top 4 features, and the NK cells and CD8 T cells are starting to merge in with the CD4 T cell island over here, whereas they're still sort of well separated over here. And you can see we've taken away about 8 features, and we're still getting a reasonably good map over there. And here's another way of sort of looking at it. So this is heat for CD8, and we've taken away no features. Now we've taken away the top feature, and it's already starting to fall apart. 4 features, 13 features, and I was really struck by how there's still some structure at 13 features or 16 features here. So we've essentially totally lost all the structure here, but yet we've still got a little bit of an island for CD8s off on the corner there. So MEM is good at identifying markers that are useful for clustering. Does it identify anything important? Does it find what biologists find? So this is from the Nature Immunology article from Evan Newell's group, and one of the things that they discovered was this innate lymphoid cell population number 28 that they came to call CD11b intermediate ILCs. And so if we compare the MEM label within the ILC populations for all of these, this population's top feature is CD11B enrichment, but it's only +5. And if we look at other populations that express CD11B, they're +10. And so one of the things we like about it is that it's not just high or low, it actually puts a number on it that's quantitative. We could even do decimals if we want. And we can sort of reanalyze people's data. And so this has been a dream of mine for a long time, that we could go back to published data and reanalyze it and start to get what was defining cell identity and learn new things from published data. And so here we took neutrophils from all the different tissues and compared how their MEM scores differ across tissue. And the take-home from this is that even when cells fall in a kind of the same part of the VISNI map, they can have really different MEM scores. And so the lung neutrophils were very high for CD54, and they were one of the only populations that was enriched for CD54, and that's part of their trafficking to the lung. So the other key thing is that we get pretty much the same MEM label for the same type of cell across different tissues and across different cytometry platforms. So we've compared fluorescence and mass cytometry for CD4 T cells and B cells. And so this is showing just a table from the paper of the MEM labels for CD4 cells. And you can see CD4 is usually one of the top features of CD4 T cells. In this case, we expert gated for CD4s. And so of course it's going to be there, but we're also typically pulling out a little bit of CD3 enrichment in them and some other things. You can see other elements of the populations vary. Even in a panel focused on B cells, we were able to characterize the CD4 T cells well, because this was mass cytometry. And so in this figure, what we did is we took only the MEM label, and we asked, if we compare the MEM labels to each other quantitatively, do we align the MEM label? How similar are the MEM labels? For those who are really interested, we took each number in the MEM label and did a root mean square deviation of it versus the other population. So just using the text MEM label, can we classify populations from, I think it was 7 different studies over time, from mass cytometry and fluorescence cytometry. So we went and we expert gated for CD4 T cells or B cells in these studies. In some cases, so pink means that it was fluorescence flow, gray is mass cytometry. The dot means that it was stimulated with SEB, so we had one whole experiment where we had stim and unstim, and then the shape, which you can't really see very well, is either peripheral blood, tonsil, or bone marrow. And we picked these because CD4s would be pretty much the same between all of those tissues. And so you can see one of my favorite results is that fluorescence and mass cytometry clustered closer together than this data set of CD4 T cells from mass cytometry. So it was kind of pulled out from these other two. So the MEM label is working in a cross-platform way, in part because it's sort of so simple and so dumbed down that and it's a relative label, so it, I think, accounts for some of the platform-specific differences in the scale and other things. But it's very dependent on people getting their data into a good place before we calculate the MEM label. So on an appropriate scale, well compensated and all of that. But so this is the hope, is that we could now create a platform-independent database of what things we have seen expressed in particular types of cells. And if we had this database, we could now drop in a new sample of cells and figure out whether it's a CD4 T cell, a B cell, or something else. And so we can get a similarity score. So the other thing, we tried to challenge MEM. And so this was our glioblastoma samples. And this is just 4 different individual tumors, but many populations from them. And we tied our hands behind our back because we had a CyTOF panel with 30 markers, but we only looked at things that would be expressed on the cancer cells. And you can argue that we're cheating a little because we've got HLA-DR and CD44, but they're definitely expressed on cancer cells in glioblastoma, melanoma, and others. And so you can see this is the MEM label clustering that we got. And we went through these populations and we said who we think they all are. So these ones with high S100 beta, we were pretty sure that those ones were cancer cells. Over here, for example, with HLA-DR and not having S100 beta, we were thinking maybe these were antigen-presenting cells. And then these ones here that didn't have any of them. And we kind of knew what the distribution of cells should be in these. And so we weren't operating in a total vacuum. And so we guessed that these might be our CD3 T cells. And so keeping in mind that we only did the clustering with these particular features, we added back these ones and looked, and we were right. So these populations express very high CD45. These ones have some CD3 in them. These had CD64. And so without the key markers of cell identity, MEM and the computational tools that we're using are able to identify cells. And so we could start to, we hope, stitch together cell identity across data sets where even the same markers haven't been measured, but some markers are in common between them. So I think we're almost out of time. Maybe I'll give one more, I think I have one more example. So this is, MEM is especially good at these kind of challenging subsets within a complex tissue. So this was a study where we wanted to get at myeloid-derived suppressor cells in the blood of melanoma patients. And so I'll summarize a lot of data. This is one patient, lots of populations identified computationally within one patient's peripheral blood. All of our markers for our MDSC-focused panel, and this is the MEM heat map. And the population in red are 4 clusters that we decided were myeloid-derived suppressor cells. And it's really good at highlighting things like S100A9, CD13, CD32, that are differentially enriched versus other myeloid lineage cells that are present within this tissue. And so this is one of our easiest ways of picking out MDSCs now, and we're able to show that they're enriched in the blood of this melanoma patient. And our favorite marker for finding them now is S100A9, which is really, really highly expressed and enriched. So I'll just kind of mention it works for phosphoflow and fold change and can characterize changes over time as well. You can apply it, this is kind of it's a very old data set. For those of you who know the DLBCL, GCB, ABC classification, we ran MEM on patients, not on cells within those 2 groups, and looked at genes. And so this is an enrichment score for these genes within the ABC or GCB group. And some of the things in green or blue are things that have since been shown as key elements of that particular signature. And coming back to the case study that I talked about, so if somebody shows you a graph like this, we can now pop up a view through and ask, is the phenotype or the identity of that population changing over time? So this blast curve kind of implies that it's the same blast, but we can actually see that they're losing some of the features that defined them at the pretreatment stage as they're blasting off. So the highlights of MEM are we need tools that can learn and compare cytotype, which is our new word for cell identity. So we're trying to do cytotyping to cytomics as genotyping is to genomics. It creates these automatic labels that a human and a computer can read. It can characterize different cell subsets, and we're hoping that if we get lots and lots of data and stitch together MEM labels from different experiments, different platforms, we can achieve this kind of deep knowledge about cytotype. We might run all CD markers and transcription factors on sorted cell subsets. And so I'll conclude by thanking my funding sources, my wonderful lab. Kirsten Diggins is now coming on as a postdoc for a year in our lab. She created MEM, and we're open to collaborating widely around the world. The more data we can feed into it, the better. And so if you have large sets of annotated flow cytometry data you'd like to run MEM on, please contact us. We'd love to do that. And then some of the other people, so Dion did melanoma work, Nellen did the glioblastoma work, Ali did the systems immune monitoring study, and I didn't talk about the other people's work very much. Oh, Mikkel did the myeloid-derived suppressor cell work. So thank you very much, and if I have time, I'll answer a few questions. Yeah?
Very elegant talk, thank you. I wonder if you could comment on this other technology that Vandel is developing, MIMI, and compare and contrast that with the fluidized tissue mass spec and related to dissociated cells as opposed to.
Yeah, so the question is about these new imaging mass cytometry platforms. And so Baron Bodenmiller worked with Fluidigm to develop what they call IMC, imaging mass cytometry, which plugs into the flow-based Helios instrument. And then Sean Bendel and Harris Feinberg have started this company, IonPath, to commercialize MIDI, multiplex ion beam imaging Both of them scan over a tissue with a laser, ablate the tissue, and quantify the metals in a histology setting so that you can get a 35-dimensional image with the same metal-labeled antibodies that mass cytometry does. So I haven't worked myself with either of them, but I've looked at lots of data from both of them. And my understanding of them is that maybe they say they've gotten it much faster, and because it works in a vacuum, it's more sensitive, is what I've heard, but I don't know if it's commercialized yet, and my understanding is that it'll be more expensive. And I'm not a spokesperson for either group, but so I want them all, and I haven't used that. I think the imaging information, we need that, we need RNA expression, we need sequence, and we need protein and phosphoprotein, and we need all of that to get at cell identity. And so I think it's going to be critical to have some platform like this, but they're very slow compared to other imaging platforms. But I'm excited about them. Yeah.
Two questions. First one, is the memo code published? And then the second one is, presumably you use this with regular flow forging of fluorescent state in combination with single cell. Does that work as well as you do these types of computations?
So yes and yes. So MEM just came out in January, and the code is online, free for academics, up on the Vanderbilt website. Email me if you can't find it, and we'll send it to you. We've also designed a course that we're teaching on MEM, and so we're trying to spread it far and wide, because it only works if everybody adopts it and starts sharing MEM labels and using MEM as the language for describing things. So we would love for people to use it, and absolutely. So fluorescence flow, if you have well-annotated repositories of FCS files, either heme path data or old sorting data or whatever, we can now go back and calculate MEM labels on all of that and start to compare those to what do we get by other technologies. So yes, I'd love to do that.
Trying to do is in the initial sorting and then single cell RNA-seq on that.
Yeah, exactly. So if you get a sorted pool and do RNA, or I would say also run like a BD lyoplate or something and get 300 CD markers on all of them. Yeah, I know. So he made this gesture, which is the, if only we had all the money. But I think we're all going to start doing this across different areas. And the key is to run 2 platforms on each sorted thing, not just one, so that we can start to align across them and ask which things are robust independent of platform. Is CD3 the transcript that you need to include in all of your RNA-seq experiments, or what? More, thank you.